Hello, everyone, and welcome to today's live broadcast, Fast, Cost-Effective, and High-Throughput Solutions for DNA Assembly. I am Michelle Ashton of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Labroots and sponsored by Beckman Coulter Life Sciences. For more information on our sponsor, please visit www.beckman.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenters, Soheila Beck, the Echo Commercial Product Manager of Genomics at Beckman Coulter Life Sciences, and Tom Speedy, the Field Application Manager for Synthetic Biology at Integrated DNA Technologies. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Soheila and Tom, you may now begin your presentations.
Thank you, Michelle. Hello, everyone, and thank you for attending this talk. Today, we are going to talk about fast, cost-effective, and high-throughput solutions for DNA assembly. But let me first give you a quick introduction to our company. Beckman Coulter Life Sciences was founded by Caltech professor Arnold Beckman in 1935 and joined Danaher Corporation in 2011. We make and support a range of solutions from liquid handlers to cell counters to centrifuges and flow cytometers. Along with that, we manufacture and support a range of high-quality reagents and consumables to ensure delivering high performance in a variety of assays from genomics to cell viability and flow cytometry. We also manufacture a variety of labware, including tips and plates. Now, focusing on the topic of today's talk, which is setting up gene assemblies. As we know, these techniques play an important role in a lot of applied fields. For example, in faster vaccine development for rapidly evolving viruses, or generating new antibodies for therapeutic purposes, which, as we know, are some of the very hot topics these days. However, as we know, making a successful product through using gene assembly techniques involves trying thousands of different combinations of gene fragments coming together through different gene assembly methods. Some of the common methods being used are Golden Gate, Gibson, NAB Builder, or other commercial and custom methods. But no matter which method you use, as we discussed, thousands of different combinations need to go through a high throughput cycle of build, design, test, and learn with many points of failures. Therefore, it would be essential to have a solution which can reduce the assembly and QC costs and also transfer reagents and fragments from any well in the source plate to any well in the destination plate rapidly. The good news is Beckman Coulter Life Sciences offers an acoustic liquid handler called Echo 525, which is a tipless and non-contact liquid handler. Echo transfers microliter to nanoliter range of volumes down to 25 nanoliter very accurately and precisely. It has a fast flow rate of up to 6 microliter per second. So what that means, for example, is to transfer 2 microliter from a 384 source plate to a 384 destination plate, it takes less than 4 minutes. Because of these fast and accurate transfers, Echo enables reduced assembly and QC reaction volumes, and therefore reducing the cost while maintaining data quality or even improving them. So how does the Echo work? The heart of echo technology is a transducer that generates low-energy ultrasounds, which are focused on the meniscus of the liquid in the source well to shoot 25 nanoliter droplets to the wells of a destination plate, which is inverted on top of the source plate. Echo uses a unique technology called dynamic fluid analysis, which what it means is adjusting the transfer parameters in real time for a variety of fluids. So various samples such as DNA, RNA, enzymes, master mixes can all be transferred at different concentrations without any user calibration. All user needs to do is choose the right calibration from a short list of available calibrations. Echo has a very easy and user-friendly software in which a user would upload a pick list to dictate the transfer pattern from any well in the source to any well in the destination. As mentioned, these transfers are fast. For example, a tip-based liquid handler may take 6 seconds to do a well-to-well transfer, but for Echo, this would be less than a second. And therefore, the combination of software and hardware make Echo a great fit for gene assembly workflows. Echo comes in a variety of flavors. So far, I talked about the Echo 525, which transfers 25 nanoliter droplets of aqueous fluid in the range of nanoliter to microliter. We also offer Echo 650 and Echo 655, which transfer 2.5 nanoliter droplets of both DMSO and aqueous solutions, and are also compatible with storage tubes, which means that you don't need to transfer the samples from storage tubes to a plate before transferring on Echo, and you can directly transfer from tubes. Echo 655 is much faster than Echo 650. Here at Beckman, we also offer modular, flexible, and scalable integration solutions called access systems for walkaway and high-throughput workflows. So let's look at a typical gene assembly workflow. Echo can help in pooling of the fragments, setting up the gene assembly reactions in a smaller volume, and even spotting the culture after transformation to get the single colonies, and also help with any optional QC steps such as colony qPCR or NGS, as well as any downstream functional testing to miniaturize and optimize those assays. For example, a TXCR reaction that requires lots of optimization. Let's look at an example. Recently, we did an application note with NEB, which is available on our website. We miniaturized the assembly reaction volumes for NEB Builder Hi-Fi kit using Echo 525 liquid handler. Fragments were ordered from IDT in Echo source plates, so they were ready to go on Echo when arrived in mail. These reaction volumes were reduced from 20 microliter done manually to 500 nanoliter on Echo. So the volumes that are reduced on Echo are shown on this table from 4 microliter down to 500 nanoliter total reaction volume. For the 500 nanoliter reaction, only 1.25 femtomole of each fragment piece was used. Therefore, there is a lot of cost saving on the reagents. After 1 hour, 50 degrees Celsius incubation, the E. coli competent cells were added. For example, for the 500 nanoliter reaction, 12.5 microliter of the E. coli cells were added directly into the PCR plate. So now we are at this step of the workflow. The transformation is done, and it's time to do the colony spotting or the culture spotting to get the single colonies. Echo can be very helpful in spotting these cultures in a dense format and save on a lot of LV agar Petri dishes that would have been generated otherwise. Did you know that Echo can create masterpieces? Folks at Jeff Boca's lab at New York University have used their Echo to create these beautiful art pieces by dispensing colored yeast on SBS LV agar plates. You can see more amazing work like this on eastart.org. Now back to our workflow, we are at the QC step, which can be optional in some high-throughput labs to do either colony qPCR or NGS library prep. So Echo can be of great help miniaturizing and automating colony qPCR setups. The colonies are lysed in lysis buffer in an Echo source plate. Primer and probes can be ordered in Echo source plates from IDT, and master mix will be dispensed from a reservoir source plate. Then Echo will set up the qPCR reactions in a fraction of volume and therefore cost, and then data will be analyzed. A typical data from an experiment like that is shown here. You can easily tell from the amplification curve whether a junction is missed and not formed, or if the construct has been successful like the one on top. So the colony qPCR can be a very fast and cost-effective way of QCing the gene assembly workflows, as you know, and Echo can make it even more cost-effective and faster. To look at a typical cost saving for such workflow offered by Echo, this is an example from one of our customers. They reduced their reaction volumes from 10 microliter to 1 microliter total volume of qPCR reaction, and therefore reduced their reagents volumes accordingly 10 times. Between the savings on the reagents and tips, they had 8-fold less cost per sample. Another way of doing qPCR for constructs would be doing next-gen sequencing to get that single nucleotide resolution for the whole construct, for example. In a typical workflow, for example, for Nextera XT from Illumina, a traditional liquid handler, such as Biomac I-Series, can prepare the plasmid mini press, and then Echo can set up the fragmentation and PCR setups for the library prep, while reducing the reaction volumes up to 100-fold. After the PCR, some labs will do individual bit cleanups using a Biomac I-Series, and then go to one-step super fast pooling and normalization using Echo. Please note that Echo, unlike tip-based liquid handlers, can pool and normalize based on the quant data in the same step, and this will save a lot of time and save on an intermediate dilution step. Some labs decide to not do individual bit cleanup and pull directly after PCR using the Echo, and Echo can do that also really fast. The customers at Amaris Biotechnologies have reduced the reaction volumes using Echo up to 100-fold by going from 50 microliter to 500 nanoliter. An example of a cost savings is shown here by an example from one of our customers. They have been able to miniaturize their NEB builder construct building 40-fold to have 40-fold reduction in cost from $18 to less than $1. And for the Nextera XT and bead cleanup step, they have been able to reduce 10-fold. Some customers are happy with just 10-fold saving and not pushing the miniaturization any further to not worry about the environmental effects such as evaporation. So it's a decision of the lab. Another great example of Echo being used in common gene assembly kits to miniaturize the reaction volume is shown by a paper in Patrick Kai and his group published in Journal of Laboratory Automation in 2016. As shown in the graph on the left, the Gibson assembly reactions were miniaturized from 20 microliter being set up manually as control to 1 microliter down to 50 nanoliter using Echo. The interesting thing to see is the assembly efficiency has actually increased going down from 20 microliter to 1 microliter, and it stays the same at 500 nanoliter with the 20 microliter. So Echo not only improves the assembly efficiency by miniaturization, but it can also offer cost savings. Another example is the Golden Gate assembly shown on the right, where the reaction volumes were miniaturized from 7.5 microliter being set up manually to 1 microliter and below down to 50 nanoliter on Echo. And as you can see, the assembly efficiency has maximized at 500 nanoliter reaction assembly, along with the cost savings that it offers. So this is not uncommon to see that miniaturization of the reaction volumes using echo actually improves the efficiencies and data quality. Authors mentioned that we envision that acoustic dispensing will become an instrumental technology in synthetic biology. So far, we talked about the cost savings of echo through miniaturization of reaction volumes while offering even better assembly efficiencies. But now I would like to highlight the time savings that it offers. Based on data from Amyris Biotechnologies and other customers, we can see that echo can make these workflows, for example, 15 hours faster. So for example, in a traditional liquid handler, on a traditional liquid handler, an assembly can take about 12 hours, and that has been reduced to only 3 hours on echo in Amyris Biotechnologies. Or a pooling and normalization can take 6 hours on a traditional liquid handler, and that can be reduced to 12 minutes. And this is data from another lab. So the time savings are also huge besides the cost savings and data quality. To summarize the liquid handlers involved in such workflows, while echo is an essential part of a gene assembly workflow to create assemblies, spot the colonies, and set up the QC reactions in a smaller reaction volumes, there is a need for a larger volume liquid handler, such as Biomax I-Series, to add the competent cells and media to do the transformation and to do any colony picking, mini preps, and optional bead cleanups. Now, Echo and Biomax both are offered by Beckman Coulter Life Sciences, so you can come to us for one-stop shop for all liquid handlers from nanoliter to milliliter and getting the application and engineering support for optimizing your workflows from one vendor. I have shown how such systems can be integrated together for more walkaway and high throughput workflows in these pictures. The gripper arm of Biomax will pass the plate between Echo and Biomax. I would like to highlight a quote by Dr. Sunil Chandran, VP of R&D at Amyris Biotechnologies. Echo is an extremely fast instrument, a significant savings in terms of time. We've been able to compress pipelines that take about 12 hours to about 3 hours. Here I have shown the access system, which is our walkaway integration solution with the robotic arm and plate hotel next to their Echo. To summarize the key features and benefits that we talked about in this talk, Echo, by going to small volumes down to 25 or 2.5 nanoliter, depending on the model, can reduce in reagent costs and conserve on precious samples. Echo is a non-contact acoustic drop ejection system, and therefore it can improve data quality and sustainability through using less plastics. It's a fast any well to any well transfer, and therefore it will reduce assembly time from hours to minutes, and it also enables fast QC. Here at Beckman Coulter Life Sciences, we provide a full portfolio of solutions, from instruments to reagents to software and great field application and service engineering support. Thank you everyone for your attention. We really look forward to hearing from you about your simple to complex projects and working with you. Now I would like to pass it on to our next presenter, Tom Speedy from IDT.
Thank you, Soheila. Thank you all for joining us today, and thank you to our host, Labroots, and our sponsor, Beckman Coulter Life Sciences. I'm pleased to present to you today on our fast, cost-effective, and high-throughput solutions for DNA assembly from integrated DNA technology. IDT was founded over 30 years ago in Coralville, Iowa, and is the global leader in DNA and RNA synthesis. We have a global operation headquartered in Coralville. We have research and development facilities in California and a production facility. We have a brand new production facility in North Carolina, and our European operations are centered in Leuven in Belgium and our Asia Pacific operations in Singapore. We also have a variety of sales offices around the world as well. We now have more than 1,500 associates and more than 130,000 active customers. And to give you an idea of the scale of our operations, we manufacture 90,000 oligonucleotides and over 1,100 genes and gene fragment products per day. This equates to over 112 million bases per month. So integrated DNA technologies' core products are oligonucleotides, and as I mentioned, we have over 30 years of industry-leading quality and cost efficiency. Our founder, Joe Walder, realized that the cost and quality of oligonucleotides at that time was very high cost and very poor quality. And therefore, he started IDT by offering free oligonucleotides to academics in return for them sending him their old synthesizers. So over the years, we have developed and optimized all our technologies, which I will come on to in just a moment. In addition to our core products, we have also built a range of other products from those for a variety of applications. These include, as we're discussing today, synthetic biology, but also qPCR, gene synthesis, NGS, molecular biology, genotyping, and various others as well. So to give you an example of some of the innovative products that we've made leveraging our expertise in DNA synthesis, here we see some examples from our prime-time qPCR probes, primers, and assays to our X-Gen lockdown probes and blocking oligos for NGS. We have a complete portfolio for CRISPR. Our Altar CRISPR reagents range from design through enzymes and guides into HDR enhancers and single-stranded templates for efficient integration. And we also have our G-Blocks gene fragment products, which we're going to go into more detail in just a second. We offer a variety of customized solutions for your oligonucleotides. You can order single-stranded or double-stranded DNA in plates or tubes. We also offer oligo pools and primer mixes, and we offer to aliquot and provide replicates if required. The IDT continues to provide customers with leading-edge genomics products as a result of our commitment to sustainable growth and the vertical integration of our products. IDT, we make all our own high-quality chemical reagents for oligosynthesis in-house. We develop our own instrument platforms and engineering solutions. We have scientists creating custom software for our manufacturing processes and our customer ordering portals. We also have a very strong team of customer care associates who provide a premier customer service experience. In fact, our Net Promoter Score is 9.12, which exceeds that of many of the world's most renowned brands. Our IDT scientists have created innovative products of the highest quality with industry-leading quality control capabilities, and we continue to develop these offerings. So now I would like to discuss the synthetic biology portfolio at IDT. As Soheila mentioned, fundamental to synthetic biology is the design, build, test, learn cycle. And one of the things that really excites me about synthetic biology is the way that numerous disciplines have come together, such as chemistry, computer science, biology, engineering, physics, to provide solutions in a range of areas such as biosensors, biomaterials, biofuels, biomedicine, including diagnostics and therapeutics, bioremediation for food, flavor, fragrance, and fine chemicals. A really great example of this is a UK synthetic biology company called Colorifics, who have developed an ethical and sustainable technology for dyeing textiles. They realized that the textile industry processes were creating a huge amount of pollution. And through the use of synthetic biology, they have been able to engineer bacteria, yeast, to fix and color textiles eliminating toxic wastes and reducing water use by over 10 times. In fact, I actually recently received some shirts which have been dyed using the colorifics technology, a purple ID t-shirt where the color is derived from silk cocoon, and a beige ID t-shirt which is color is derived from melanin from gorillas. IDT fits into this design, build, test, learn cycle, providing the tools for the build component. We have a range of products all the way through from our oligonucleotides to opals, our DNA fragments through to our clonal genes, and then our pathways group for more custom solutions, and finally our single-stranded megamers. All of these synthetic biology products are built from our ultramer oligonucleotides. These are used to assemble the gene fragments, including our G blocks, our E blocks, and the new product that I'm about to mention in a moment. We use these fragments to assemble custom genes, and we use these genes to make the single-stranded megamers. As I mentioned, we also have a custom project team who are willing to take on any projects outside those through our high-throughput manufacturing processes. So first, I'd like to introduce the E-blocks gene fragments. These have been made for speed. They typically ship in 1 to 3 business days. They're available from 300 to 900 bases, and they're priced very competitively at 6 cents per base. They're particularly suited to antibody discovery, vaccine development, high-throughput screening, and they're available in a variety of shipping formats, including the Beckman Coulter Life Sciences Echo Plate. Our original gene fragment product was the G blocks. They were the first commercially available fragments launched in 2012. These were designed for gene construction, NGS controls, and qPCR controls and standards. The real benefit in qPCR is the ability to combine multiple amplicons in a single fragment, therefore ensuring uniform concentration in your assay. And also that you can have synthetic amplicons for those orthologs that you're not able to find in nature. These range from 125 to 3,000 bases and are delivered in 2 to 8 business days. Again, these are available in a variety of different formats. The final product I want to mention before moving on to the new product is our custom gene synthesis service. We offer custom clonal genes from 25 to 5,000 bases. These are NGS verified and available in either a standard IDT vector or your own custom vector. One thing to mention is that there are no hidden cloning fees when you order our custom gene synthesis services. So I'm very pleased to announce the imminent launch of a new product in our fragment family range. The G-Blocks Hi-Fi gene fragments offer the industry's lowest error rate, 1 in 12,000. They are double-stranded DNA available from 1,000 to 3,000 bases and delivered in 6 to 10 business days. These fragments are NGS verified. So just to summarize the products in our fragment family, we have the E-Blocks designed for speed, which are particularly suitable, as I mentioned, for antibody, protein engineering, vaccine development, or high-throughput screening. We have our G-Blocks gene fragments, which are available in tubes or in plates. These are particularly suitable for gene construction, qPCR standards, and NGS controls. And we also have our G-Blocks Hi-Fi gene fragments, the newest product to the family, which are the highest quality fragments and suitable for large gene construction, including pathway development or microbe design. One thing that's universal across all of the fragments within the family and with any synthetic DNA fragments is the potential issues when synthesizing these, and there are a number of complexities that should be avoided. In particular, things like secondary structure, hairpins, or extremes of GC, repeats and homopolymeric runs. And this is particularly important near the terminal ends of a sequence. At IDT, we offer a variety of solutions for the optimization and design of your double-stranded fragments to ensure fast and high-quality delivery. So the G-Block's Hi-Fi fragments show the lowest screening effort to find the correct clone. We found that there's a greater than 90% chance of finding the correct colony when using the Hi-Fi fragments. As you can see here in this table, a comparison between the new Hi-Fi fragments, other suppliers, and the products already within our portfolio. So here's some data showing cloning success with the G-Block's Hi-Fi fragment. As you can see, the predicted cloning efficiency on the left, as the length increases, the predicted success decreases. But because of the exceptionally low error rates in the production of G-Block's Hi-Fi gene fragments, the predicted success of cloning is higher than when similar other fragments are used. On the right, you can see that the G-Block's Hi-Fi gene fragments offer over 90% cloning efficiency, while other fragment products are below 80%. So just to summarize, the G-Block's Hi-Fi gene fragments designed for large gene construction, pathway development, or microbe design are the highest fidelity and the purest double-stranded fragments available in the industry. You pick 50% fewer colonies to get your gene of interest, and they're flexible for all downstream cloning methods, including Golden Gate, Gibson, restriction cloning. There are no universal linkers to remove, so they're ready to use, and they ship dry or normalized in tubes or plates, including the Beckman Coulter Life Sciences Echo plate. So here is an example of how those fragments are assembled using the Acoustic liquid handler. So in this example, double-stranded DNA fragments were designed with overhangs, and then using the New England Biolab's Any Builder Hi-Fi DNA assembly cloning kit, you're able to assemble those into a large construct. The basic principle is that the proprietary 5' exonuclease digests the overlaps and creates overhangs. The complementary 3' overhangs are then filled with a DNA polymerase upon annealing. DNA ligase is used on the final product to seal nicks and create a continuous assembly. The entire reaction is incubated at a temperature of 50 Celsius for as little as 15 minutes in a recommended volume of 20 microliters. The synthetic biology products and integrated DNA technologies are assembled with the Acoustic liquid handler, the Echo 525. This allows us to rapidly deliver E-Block's gene fragments in as little as 1 business day. It reduces the reaction volume by as much as 48 times versus traditional liquid handlers. And as I mentioned previously, sustainability is key to synthetic biology. It's a big focus for a lot of the companies and the researchers active in this space. And the Acoustic liquid handler saves us over 3 million plastic tips per year. At Integrated DNA Technologies, we're committed to minimizing the environmental impacts of our manufacture and research operations and our products and services, and we're continuously working to improve those through the applications and use of new technologies like the Beckman Life Sciences acoustic liquid handlers. We've also integrated a number of sustainable features into our headquarters building in Coralville, Iowa. This includes geothermal heating and cooling, use of recycled building materials, the application of energy-efficient equipment, and solar shading to allow natural light without increasing heat load. We also use stormwater bioswales, prairie grasses that naturally filter the parking lot runoff. So to summarize, IDT is a complete product line to support synthetic biology research. We have a history of sustainable automation of high-throughput workflows. And coupled with the Beckman Coulter Life Sciences Acoustic liquid handler, this is an enabling technology that allows us to supply our customers worldwide with consistent quality products and with reliable turnaround times. And we're very pleased to announce the imminent launch of the new G-Block's Hi-Fi gene fragments that are perfect for a wide range of applications and will hopefully drive synthetic biology forward over the coming years. Thank you very much for your time today. I look forward to questions. Thank you.
Thank you, Soheila and Tom, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question is, does the echo require calibrating for each fluid type?
Echo has some preset calibrations, and all user needs to do is pick one of the preset calibrations that is a short list. So no user calibration at the machine is required or even possible.
Great, thank you. Okay, next question is, what volume are G blocks resuspended in when shipped in Echo 384 well plates?
Thank you, that's a good question. We resuspend our G blocks hi-fi fragments in a volume of 50 microliters at a concentration of 20 nanograms per microliter. This is true for the Echo 384 well plates and also for our other shipping formats for tubes and our 96 or 384 well PCR plates.
Thank you for that. Okay, now this next question asks, can you use any type of plate with the Echo?
You can use any destination plates that are SPSS standard and automation friendly, of course, on Echo, so that's really a wide range of plates. On the source plates, because we want to ensure of great quality of transfers, we require users to use our Echo source plates that are a variety of plates available to the users.
Thanks, Soheila. Now, Tom, I think this question's for you. Can I amplify my G blocks if I need more yield?
This is a question I get quite often, actually. So for most applications, the material that we provide is ready for use and at a sufficient yield. So PCR amplification shouldn't really be necessary. However, if you do wish to amplify G blocks smaller than 1 kb, standard G blocks, then we would recommend using the minimum amount of starting template required by your PCR kit protocol and limit the cycles to 10 to 12 using a high fidelity polymerase. If you have sequences longer than 1 kb, then we strongly recommend you purchase the G blocks hi-fi fragments and follow the same procedure. These are designed really for assembling, they're much more designed for amplification and for the assembly of very large constructs due to the very low error rates.
Thanks, Tom. Now, how do I design my G blocks for restriction cloning?
Okay, yeah, when you're cloning G block gene fragments, it's very similar to cloning a very pure PCR product. One important thing to remember is to add 6 to 8 bases at the ends of your fragments. But on the restriction recognition sequences, most enzymes won't cut efficiently, if at all, if the fragment terminates precisely at the restriction site. So do remember to add those extra bases in your design.
So Soheila, let's go ahead and move over to you. How does the ecosystem ensure there is no cross-contamination?
That's a good question, and that's the one that we get often. So between the source and destination plate, that, as you know, destination plate is inverted on top of the source plate. Between them, there is only 0.5 millimeter gap, so they are really close to each other. And the drops are transferred to the center of the destination plate. So when users choose the right calibration, which is a wide calibration, it's not just specific to a specific liquid. It's a wide calibration covering wide range of liquid properties. The drops end up in the center of the destination plate, and that's how we ensure of no cross-contamination.
Now, if the destination is inverted, why doesn't the liquid fall out of the wells?
That's another common question we get. Thanks for asking that. So I know it may sound a little strange, but surface tension does wonders, and the liquid actually sticks to the destination plate upside down up to a large amount of volume. For example, with water or with solutions that have high surface tension like glycerol, we can hold upside down maybe 40, 50 microliter depending on the coating on the destination plate, but those are not uncommon volumes that we hold upside down based on surface tension holding the liquid. But now if you have detergents, that volume will reduce.
Great, thank you. What length of overlap region do you recommend for Gibson assembly?
I'm guessing that one's probably for me. So yeah, we generally recommend overlaps of between around 30 to 80 base pairs for Gibson. In general, using overlaps longer than 35 base pairs is recommended for larger constructs, particularly those greater than 3 kb, or where you're assembling 3 or more fragments. We have a full Gibson assembly protocol available on idtdna.com, and also New England Biolabs NEB Hi-Fi builder kits also have a very specific protocol that will help you with that.
Can the echo be used to transfer live cells?
Absolutely, and this is also a common question. Echo uses very gentle ultrasounds, and the drops are big enough that cells actually fit in a drop, and we do a very gentle transfer of the cells, and those are some of the very common applications that echo is used for.
Great, thank you. Now, how long would it take to transfer 2 microliters from a 384W plate to another 384W plate on echo?
To transfer from a 384 source plate to a 384 destination plate one by one, from let's say A1 of the source plate on one plate to another well in the destination plate one by one from each well, it will take only 2 minutes on Echo 525.
Great, thank you. Now we have time for just a few more questions. Tom, this question's for you. Can I directly transfect G blocks into mammalian cells?
That's a great question as well. Yeah, these are really good questions. So G blocks and the G blocks Hi-Fi gene fragments are suitable for direct transfection. There's a few things to consider though. We do recommend filter sterilization with a 0.22 micron filter before use. Some researchers have observed toxicity in cells when you transfect large amounts of double-stranded DNA. That sensitivity does vary depending on which cell line you're working on. So we do suggest titrating concentrations of the double-stranded fragments just to determine what that optimal delivery will be for your particular cell type.
Great, thank you. And our last question is, how many colony spots have you tried spotting on the SBS Omni trays?
That's a good question. So we safely transfer 1536 spots on an LB agar plate that is a standard size, like those Omni trays you mentioned. But it doesn't mean that if we push for it, we cannot go higher, but 1536 is a safe number.
Thank you again, Soheila and Tom. Do you have any final comments for our audience before we go?
I wanted to invite the audience to feel free to reach out to us with any of their simple to complex gene assembly projects and let us be part of their solution.
Yeah, absolutely. As Soheila says, please do contact us with any questions or if there's anything we can do to help. And I'd just like to say thank you again to everybody for joining us today. and to our host, Labroots, and our sponsors, Beckman Coulter Life Sciences. It's been a pleasure speaking to you. Thank you.
Before we go, I'd like to thank the audience for joining us today and for their questions. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to once again thank Soheila and Tom for their time today and for their important research. We would also like to thank Labroots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.